Welcome to today's lecture about parasites, epigenetics, and chemosignals. I want to start today's lecture with a story about a woman named Susanna Callahan. In 2009, Susanna Callahan was 21. She was living in New York City. She was a journalist at a big newspaper. She had a boyfriend. She had her own apartment. She was enjoying her life. One day, she was at her boyfriend's apartment, and this sudden paranoia overtook her. She decided when he wasn't home, she had to go through all of his stuff and take out boxes from his closet and analyze his entire apartment. And this behavior was atypical for Susanna. She later went home and suddenly became paranoid that she had bed bugs. She felt like she was itchy all over her body, and she felt like she saw these bed bugs all over her apartment, and so she put all of her belongings in garbage bags. She then went to work, where she worked as a journalist at a newspaper, and she started feeling nauseous, and she had paralysis in one of her hands, or numbness in one of her hands. And slowly, as the days went by, her mental state deteriorated. So her work deteriorated. She was feeling extremely anxious and nauseous, and the numbness got worse. She kept feeling this jealousy and this irrational paranoia about the bed bugs. And finally, about 2 weeks after the first incident of going through her boyfriend's belongings, she had a seizure. When she had a seizure, she was brought to a hospital. And in that hospital, she stayed for 1 entire month. So at the beginning of her hospital stay, she gradually got worse and worse. Her parents were extremely worried about her, and it wasn't even sure if she would survive. She was losing her ability to speak, her memories were deteriorating, she was becoming irrational, paranoid, restless. And after a few days in the hospital, she was diagnosed with schizophrenia. So really, this is a woman who was perfectly functioning and fine, and within 2 weeks had a diagnosis or 2 weeks to 4 weeks had a diagnosis of schizophrenia. So like I said, she was in the hospital for 1 month. She had $1 million of medical fees after this 1 month. But luckily, one doctor crossed paths with her, and he decided to do a few extra tests because he had recently heard of a new disorder called anti-NMDA receptor encephalitis. And he did these blood tests on her, and he found out that that's exactly what she was suffering from. She had this extremely rare disorder called anti-NMDA receptor encephalitis, which basically means that NMDA receptors, which are one of the most common receptors within the human brain, are being attacked by antibodies from the own body's immune system. So it's an autoimmune disease. So this doctor ran this test. She tested positive for this disorder. It's a quite simple procedure to help cure this disorder. Basically, drugs that will dampen the immune response, which will stop the antibodies from being produced and then lead to a full recovery. And that's exactly what happened. After a few months of treatment, Susanna became completely healthy, completely recovered, as though nothing had happened. I tell this story because this disorder that Susanna had was only discovered in 2007. So imagine how many people before 2007 had this disorder, were probably diagnosed with schizophrenia or some other disorder, and were left untreated for the rest of their lives. So since then, there's been a few thousand cases of this anti-NMDA receptor encephalitis. Some doctors believe there are as many as a few million cases out there in the world, yet they have not yet been diagnosed because it's so rare. So this is an example of a disease which affects someone which is kind of invisible. So we didn't know about this before 2007, and imagine how many other disorders there are that have yet to be discovered that can affect us and have these strong effects. So to demonstrate that, there was a study done in New York City's subway a few years ago. Researchers went into the New York City subway system and swabbed different surfaces, and then took these swabs back to the lab to analyze what kind of creatures were living in the New York City subway system. And they found human DNA, animal DNA, they found some bacteria, some viruses, parasites, and yet 70% of the DNA that they found was unidentifiable. So they didn't know what it was. Basically, unidentified subway organisms. And I'm laughing, but it's really not funny because we don't yet understand all these organisms that are out there in our environment. And it's very possible that these organisms could be affecting us and could also be affecting us psychologically as well. So the first topic I have today is parasites. Parasites are essentially organisms that need another organism to live. So they live on or in an organism and basically take advantage of them to increase their odds of survival. And some of these parasites actually hijack the behavior of the animal in order to make them more likely to survive. The first example I have is rabies. Rabies is a virus, but some viruses and bacteria can be considered parasites if they use that organism to propagate their own survival. So what happens when an organism has rabies? For example, a dog. Saliva production increases, aggression increases, and their ability to swallow is affected. So they can't really swallow, they have all the saliva in their mouth and they're feeling very aggressive. This makes the dogs much more likely to bite someone or another dog or a person. And then through that bite, the rabies virus transfers into another organism because the virus lives in the saliva. So this is one example of how the virus, so it's the rabies virus that's increasing the saliva, the aggression, that's affecting the ability to swallow. And then that is helping the rabies virus survive and move on to another organism and spread. So that's one example of how a virus or a parasite can affect behavior. Another example is a parasite called Toxoplasma gondii. Toxoplasma gondii is a parasite that's primarily found in cats. It's thought that about 50% or more of cats have Toxoplasma gondii within their system. Cats catch Toxoplasma gondii by eating raw rodents. Generally, rodents know to avoid cats. However, when a rodent is infected with Toxoplasma gondii, they actually become attracted to the scent of cat urine. And so by approaching the cat urine, they then become more susceptible to be eaten by the cat. So this is one example of how the parasite Toxoplasma gondii increases the likelihood to be eaten by a cat, and it's affecting the behavior of the rodent to be attracted to the smell of the cat urine. Toxoplasma gondii is not just found in rodents and cats, it's also found in humans. It's thought that about 30% of humans are infected with Toxoplasma gondii, and that 30% mostly consists of cat owners since they have the most exposure to cats. So Toxoplasma gondii also affects behavior in humans. It's been found that if a human is infected with Toxoplasma gondii, they have about a 3-fold increase in their likelihood of developing schizophrenia. This is primarily through Toxoplasma gondii infection in childhood. So once you have Toxoplasma gondii within your body, you have it for the rest of your life. Your immune system isn't able to kill it. And so it increases the odds of developing schizophrenia. In human patients who have schizophrenia, it increases the odds of a suicide attempt. It's also been found to affect fear. So people with Toxoplasma gondii generally have a heightened sense of fear and less novelty seeking. They also have been found to have slower reaction times, not by much, but still enough that it's thought that people with this parasite actually have more car accidents. So that's been found, that people who have car accidents, then taking their blood and analyzing whether they have Toxoplasma gondii or not, they're more likely to have Toxoplasma gondii. And it's thought that they're more likely to end up in car crashes because of this slightly slower reaction time. It's also been associated with workplace accidents, again associated with slower reaction time. So this is a mysterious little parasite that's affecting cat behavior, rodent behavior, and also seems to be affecting humans in a big way. And a lot of humans appear to have this parasite within them. Another way parasites can affect us is through affecting our social behaviors. So in some studies analyzing human behavior after a vaccine, so this is after basically getting a small dose of a virus, it's been found that sociality increases. So there was one great study where they analyzed people's interactions with others. So they asked them how many people they interacted with totally in a span of 2 days, right before they got a vaccine, 2 days after they got the vaccine, and then 1 month later. And they found that in the 2 days after they got the vaccine, so that small dose of a virus within their body, that they interacted with double the amount of people as they did either 1 month later or 2 days before the vaccine. Furthermore, in other studies where people were given a vaccine or a placebo, when they got a real vaccine with a real dose of virus, these people seem to have a higher social motivation. So they claimed that they wanted to socialize with others more and spend more time with others versus alone. So this is thought to be due to the virus, which is affecting behavior and social motivation. So if you're more motivated to interact with other people and you end up interacting with other people and you're contagious, so you're not contagious after a vaccine, but it's thought that this same thing likely could happen when you're actually infected with a virus, then that virus is likely to spread to many more people while you're still asymptomatic in those 2 days before you develop symptoms. So this hasn't been studied with actual viral infection, but if the same thing would occur after being infected with a virus, so you can imagine the coronavirus, it's possible that after the infection, while you're still asymptomatic for a few days, that your social motivation actually increases before it decreases. So of course it decreases once you're actually feeling sick and coughing and not feeling great. But in those 2 days while you're still asymptomatic, it's possible that your social motivation to interact with others increases due to that virus infection, and then that makes it more likely that the virus can go and infect other people, which is pretty wild. Moving on to the next topic, epigenetics. So we all know about genetics and the genetic code. It's how you and your family are related. Our DNA is in every cell of our body. However, it's our epigenetics. So epigenetics basically refers to which genes are being expressed within their body. And epigenetics are responsible for many things. For example, how twins are different from each other. When twins are born, their epigenetic code is actually quite similar, and yet as they age, they become more and more different. And epigenetics are basically affected by the environment. So your diet, the experiences you've had, exercise, whether you take drugs, etc., etc. So epigenetics explain the differences in twins. They also explain the differences in your own cells. So for example, I have skin cells, I have blood cells, and those cells are very different. I have neurons. Those cells are different from my blood cells and my skin cells, but they all have the same genetic code. So my DNA is the same in every cell as is yours. However, it's due to epigenetics that basically turn on certain genes to make some cells skin cells, some cells neurons, some cells blood cells, etc. Actually, a similar process occurs with bees. So interestingly, every bee within a hive has the same genetic code. So they're essentially clones. And it's only through epigenetics that differentiates which bee is a worker bee or a queen bee and all the other types of bees. But within one hive, they're essentially clones and have the same genetic code. So all these examples goes to show how much epigenetics actually have an effect on us. So now you're probably wondering how epigenetics works. So I have a quick example. So DNA is often related to zippers because DNA unzips. So imagine this is a zipper. DNA is twisted up. It's a double helix, right? So it's twisted up. And when DNA essentially unzips, first it untwists, and then, I have to get the zipper. It unzips. So imagine this is DNA, the pink zipper. Then RNA comes along, and RNA reads the DNA. So it'll slowly go down the genetic code, the DNA. Okay, so imagine it read this part of the DNA, and then the RNA goes, and from this RNA, which is made from reading the DNA, it then goes to make proteins. So maybe this codes for 5 different proteins. And this is essentially how our body works. Our body needs these proteins to constantly be made and affects everything within our body. So the RNA reads the DNA, and then from the RNA, proteins are made, okay? So what epigenetics does is imagine this is the DNA, and we have something called a methyl group. So a methyl group sits on a part of the DNA. Okay, so imagine that's a methyl group. Then when the RNA is trying to read the DNA, so it's here, reading the genetic code, once it hits this methyl group, this system doesn't work anymore. It can't read the DNA, okay? So basically, the RNA will stop here and continue here, but it won't read this area, meaning that the area that would have been here on the RNA is not there, and no proteins will be made for this gene. So maybe this could be a gene related to the oxytocin receptor or something, and then this will affect whether certain proteins will be made. So that's what methyl groups are. There's another way that epigenetics works. So you could think of the methyl groups as basically on and off switches. So if a gene has a methyl group on it, it's basically off and won't be read. And histones, hold on, I need to rezip my genes. Okay, so like I said, DNA is normally twisted, so it's that double helix pattern. So imagine normal DNA is twisted like this, and then it untwists and unzips. So with histones, that affects basically how tightly the DNA is coiled. So if you have a histone group within the DNA, it'll basically be much more tightly coiled. So imagine really tight. And then it's less likely to unzip and then be read. So it's not impossible. It will sometimes be untwisted, but it's not as likely. So you could think of histones as sort of the volume. So, basically how tight it's twisted is kind of like the volume of how much is going to be expressed. And then the methyl groups are like the on and off switch. And epigenetics don't just affect us, they could actually be passed hereditarily. So there's been plenty of studies in animals to show this. So for example, if a mother is stressed during a pregnancy, then this stress during the pregnancy, and I don't mean one stressor like getting stuck in traffic once, I mean like a really strong stressor, like being in a war or being in an abusive marriage or something like that. So a strong stressor while pregnant, then this will affect the DNA of the child while the mother is pregnant. And this basically epigenetic change, because of the stress of the mother, can then be passed actually to her grandchildren. So that means if your grandmother experienced a strong chronic stressor while she was pregnant, that epigenetic signature of that stress could still be within your genetic code epigenetically, and this could affect how you now respond to stress, which is pretty wild. So moving on now from epigenetics to chemosignals. What are chemosignals? Chemosignals refer to chemicals within our environment that affect our behavior, especially chemicals coming from other humans. So you've probably heard of the term pheromones. Pheromones are generally referred to in the animal literature. So things that affect attraction between animals or increase aggression with animals. So in humans, we don't really use the term pheromones, but we refer to chemosignals. And in the past about 10 years, or 15 years, there's been quite a big surge of chemosignal research within humans. So for example, studies have shown that if you get sweat from people who are experiencing fear, and then have people smell this sweat, so it's usually within some cotton pad, then they experience higher vigilance compared to just normal sweat of people who weren't experiencing fear. And in an fMRI scanner, it's been shown that smelling this fear sweat actually increases activity within the amygdala, which is a region known to be associated with fear, versus just smelling normal sweat from someone else. Another study analyzed stress sweat. So stress sweat releases different compounds versus normal, like say exercise sweat. So they collected stress sweat versus exercise sweat from people wearing t-shirts, and then they had actual dental students smell either the exercise sweat or the stress sweat, and then they had to do a dental exam. And the dental students who smelled the stress sweat actually performed worse than those who smelled the exercise sweat. And what's interesting between these sweats is that people can't really tell them apart. So when asked, like, does this smell worse than this one, or can you tell a difference, it's completely unconscious, and it's not like you smell these chemosignals and immediately know what it is. But that's another example of how it has affected behavior. There's another study that looked at boxers. So they collected sweat from people either doing exercise or boxing, and the boxing was thought to increase the aggression within the people doing the boxing, and then had people smell the exercise sweat or the boxing sweat. And people who smelled the boxing sweat actually experienced higher levels of anxiety. And it's thought that this was basically sweat associated kind of with aggression, and by smelling chemosignals associated with aggression, that then increased anxiety in the recipient who was smelling it. So another example of how these social signals can be transferred within sweat. Finally, there was a study that analyzed people's ability to smell, so basically whether people were better smellers versus worse smellers, and then their social network size. And it's been found that a better nose is associated with a larger social network. And this again supports this idea of chemosignals. So if people have a better nose and are maybe better able to smell chemosignals within their environment or when they're interacting with others, that might then make them better social agents and make them better able to manage a large social network if these chemosignals really are playing an important role in our social relationships. So that's the end of today's lecture. The point of this lecture was kind of to teach you about different things that could affect our behaviors and our cognitions and our emotions that are kind of invisible and we don't really completely understand yet. So hopefully you have a better respect now for how mysterious human behavior is and how much there is left to find out. See you next week.